Chapter 7 starts Unit 2 for us. Chapter 7 is all about biotechnology. So basically the application of all of this information that we have learned over the last 50 years or so about DNA and about gene expression and how things work. So we're going to talk about some of the cool ways that we can use DNA technology. And then once we get to chapter 8, we'll actually talk about the structure of DNA and gene expression and things like that. But for now, we're going to talk about what we can actually do with DNA. So we're going to look at the basic tools for biotechnology, look at some of the ways that biotechnology helps us in the pharmaceutical industry to treat disease and to prevent disease. We're going to learn a little bit about genetic engineering, recombinant DNA, technology, PCR, transgenic organisms, genetically modified organisms, and then we're going to talk about some of the strengths and weaknesses of personalized gene therapy. So let's start out by defining biotechnology. So biotechnology in a general sense is just using technology to modify organisms or their cells. And this allows us to use organisms in a way that is beneficial to us. So we can basically exploit the genetic processes in a variety of organisms to give us things like insulin and forensic science and gene editing capabilities. We're going to talk about genetic engineering. So genetic engineering is the manipulation of an organism's genetic material. So we can do this by adding, deleting, editing, transplanting genes from one organism to another. So basically we can use genetic information from one organism to get another one to do something for us that we want. Now it sounds a little bit scary. We've actually been doing this for quite a while, sometimes in ways that are really sort of selection without a molecular process, and then now very in-depth molecular processes. So I know it sounds kind of crazy, sometimes sounds kind of scary, but genetic engineering is not necessarily a mad scientist in a lab somewhere. So three of the big ways that biotechnology help us are in agriculture, human health, and forensic science. So in agriculture, we use biotechnology to make pest and disease resistant crops, to increase crop yields, and to enhance the nutritional value of our foods. So you can see that there are a lot of practical benefits to this for farmers, for general populations, and then of course there are some risks that go along with biotechnology. So we'll talk about that a little bit as well. And then we have a huge potential for altering human health, improved treatment of disease through more effective medicines, improved diagnosis and screening for genetic disease, things like personalized medicine, gene editing. All of these things have the potential to greatly impact human health. So far, this is sort of in its infancy. There's been sort of a limited success in human health arena as far as biotechnology is concerned, but that doesn't mean that there aren't some really important things going on. And then, of course, forensic science. So the use of DNA technology in law enforcement, criminal justice system has been really important in being able to convict and also exonerate people for in crimes. And so forensic science has been impacted as well. All right, so let's talk about sort of the general process of biotechnology. And we're going to see these processes, maybe some of them, all of them together, in the different applications for biotechnology. So the first thing we need to do is we need to chop DNA out of the species that has a trait we're interested in. Then we need to amplify it. That means that we need to take that DNA that we're interested in and make a whole bunch of copies of it, because a small amount of DNA is not very useful. Then we need to take that piece of DNA that has the trait that we're interested in, and we need to put it into something else. In general, this is going to be bacteria or viruses as sort of little carrier machines for us to copy the DNA, to produce proteins from it, and we'll talk about how that happens. Once we insert it into those bacterial cells or whatever organism we need to insert it, we need to grow them. So we need to grow the bacteria that are going to produce insulin for us, or we need to grow the viruses that are going to transport an edited gene for us. So let's talk about how we use these more specifically. So our first step, the chopping step, we need to isolate a gene of interest. So this might be a specific gene in the human genome. Say something, we need to make something that the body normally produces, like insulin or growth hormone. But it's not helpful if it's stuck in somebody's genome somewhere. So what we would do is we would extract the DNA from the donor species, let's say humans in this case, although it really could be anything. This is not limited to humans. And then we need to cut it out of the system. So we are going to, with our DNA that we have extracted from the individual, we are going to cut the piece of DNA, the gene that we want, using something called a restriction enzyme. Now, a restriction enzyme is a small protein that is found, it was originally found in bacteria. It's actually, restriction enzymes are essentially kind of an immune system for bacteria. So this is something that occurs naturally. And the way that a restriction enzyme works is that it identifies a specific DNA sequence and then it breaks it up. So let's say that a bacteria is infected by a virus, because bacteria are infected by viruses too. Everything's infected by viruses. And the way that the bacterial cell combats this is to recognize specific sequences on that virus and cut up the DNA. And then that virus is no longer affected. So restriction enzymes were discovered in the 1960s, and a whole bunch of them, dozens of them, have been isolated since then. And each one recognizes its own specific sequence. That sequence can be anywhere from 4 to say 8, 10 DNA base pairs long. And every time it sees that specific sequence and only that sequence, it makes a cut. Now, restriction enzymes make a variety of different cuts. But in any case, whatever it's doing, it's going to make a cut at a very specific sequence. So we can see down here, we've got the targeting, and so this sequence here with this overhang is what we are going to cut out, with this darker blue or blue-green being the sequence that we're interested in. So now once we make that cut, we have isolated the gene that we want from the donor DNA. So now we have that gene. It's going to do whatever desired thing we need it to do. So once we cut it out, we're going to need to make some copies of it. So the reason that we make copies of it is that in general, you get a small amount of DNA when you take it out of an organism. So if we're using humans, for example, you might take a blood sample or a skin sample or something, but you're going to have blood sample is not a good option. Let's say skin sample or hair follicle, something like that. You're not going to get a lot of DNA. So even if you can cut out the piece that you really want, you still need to make more copies. So we're going to do something called polymerase chain reaction. Now, we haven't talked about DNA replication yet, the process for that, but basically polymerase chain reaction is DNA replication in a test tube. So the structure of DNA is a double-stranded molecule made up of complementary sequences, and we'll talk about in the next chapter what that actually means. But in order to do polymerase chain reaction, or PCR, we need to first pull apart the DNA that we want, and we do this using heat. This is called denaturing. So we apply heat to the system and it pulls apart the double helix of our DNA strand. The next thing we're going to do is add a couple of things. One is DNA polymerase. So DNA polymerase is an enzyme that is going to replicate our DNA. So DNA polymerase will come in and add the complementary sequence to our exposed strands there. So we also need those nucleotides in there to make our complementary strand. We also need a primer. Now this primer is just a short piece of DNA that we make and put in that's going to be it's going to be complementary to a region surrounding the DNA that we are interested in. And it's basically like a little signpost or a starting marker for that DNA polymerase. So once we've added those things, we cool the system, and that allows the primer to anneal to our sequence. It allows DNA polymerase to recognize that primer and start its job. The next step is extension. So in the extension step, we actually have our DNA replication occurring. So by the end of that first step, we have, instead of, let's say, one piece of DNA, we now have two identical copies. So in theory, you could start with just one copy of the gene that you wanted and make as many copies of it as you want. Then, for polymerase chain reaction process, we repeat this cycle of heating, cooling, and extension for, say, 20 or 30 cycles. And every time we do this, we double the amount of DNA that we have. So after, in the first round, we start with one, we separate it into two, then each of those is copied into two, then each of those is copied into two. So you can end up with billions of identical copies of your target DNA sequence. All right, so we have isolated and cut out the gene that we want from our donor species. We've made lots and lots of copies of it. The next thing we're going to do is insert it into the into the genetic information of another organism. Now, typically this is going to be bacteria because bacteria are easy to work with. Many times it is E. coli. E. coli is a gut bacteria that is found naturally in most people. This is the one that occasionally we hear about lettuce or something being contaminated with E. coli and it makes people sick because there are some virulent strains of E. coli. But for the most part, they hang out in your belly all the time and they actually help you with things like digestion and producing vitamins and things like that. So E. coli is readily available. There are lots of laboratory strains that you can use. And the reason that we use bacteria is that they're easy to grow, they're easy to care for, and they do the job very well. So that DNA sample that we just amplified is going to be inserted into the bacterial cell. And when we do that, we have to put it into another piece of genomic DNA, or another piece of the bacteria's DNA. I'm sorry, not genomic. We're going to insert it into a plasmid. A plasmid is a small piece of DNA that is found in a bacterial cell. They occur naturally and they generally encode things like antibiotic resistance or the ability to utilize a particular nutrient in the environment, but it is separate from its main DNA or its genomic DNA. And we use this because they're sort of temporary molecules. We take this gene of interest that we have cut with our restriction enzymes, and then we also cut our plasmid with the same restriction enzyme. And what that does is sort of makes an overhang on each of our gene of interest and our plasmid. And then, because our sequences are complementary, let me go back here real quick. You can see this cut here. Our sequences are the same both forward and backward. We can actually insert that into the plasmid. So then we put that plasmid back into the bacteria by something called a process called transformation. And then it just hangs out in that bacterial cell. Well, now this bacteria is going to express the gene that we want. So this might be something like, as I said earlier, insulin or human growth factor, because the process for expressing the gene and making the protein is the same in bacteria as it is in eukaryotic cells. So at the end of all of this, we have produced a transgenic organism. What this means, trans means different, genic means genetic information. So this means this organism has genetic information from another organism. So it's got both its own genetic information plus this new bit that we have put in. And then finally, we need to grow enough bacteria to make it useful. So we can then use the bacteria to produce or to make lots and lots of copies of that genetic information that we want. Now, something that's kind of cool about all this is we can make basically a library of all of these little fragments of DNA that have been chopped up. So we can use this library to study different pieces of DNA, to figure out what they do, how they work, what their sequence is, or we can use them for some sort of biotechnology application. But if you chop up different bits of a particular genome and then insert them into the bacterium essentially holds that information for you. And then you can analyze the DNA in a variety of different ways, depending on what you want to do with it. During this process, you have done something called cloning. So you have cloned the gene. That just means that you have made a copy of it. And then you also allow these transgenic organisms to continue to clone your DNA. So these bacterial cells are essentially clones of one another, but they're also cloning your DNA that you need. And then you can use this bacteria to store your fragments, come back to them later. So one of the coolest new biotechnology applications is something called CRISPR. Now, I graduated with my master's degree in 2008. I never even learned about CRISPR at that point. CRISPR is, within the last few years, just becoming very popular. And it is a system for editing DNA very precisely and very accurately. Now, this is not the first editing system, but it is the first editing system that is incredibly accurate. And so it's really important. There is huge potential here for a variety of applications, from editing defective genes that cause disease to anything to designer babies. Like, there's all kinds of stuff that you could do with it. Doesn't mean that you should do it, but there's a lot of things that you could do with it. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. That is quite the mouthful. You don't have to memorize that. Basically, it's a region. CRISPR is developed or was found from a region that contains short palindrome, meaning forward and backwards are the same. Short palindromic repeats is just a region in bacterial DNA. You don't have to worry about that too much. It's important, though, because it enables researchers to modify any gene. Now, this was discovered in bacteria, just like restriction enzymes, and has a similar purpose to restriction enzymes. Basically, it is part of the bacterial immune system, And just like our immune system sort of has a memory, meaning that you produce antibodies for when you are exposed to a disease, and your immune system actually remembers the virus. So this is why you can have a vaccine for the flu and not get the flu for the rest of the season, or why you can be vaccinated for chickenpox and never get chickenpox, because your immune system remembers that you were exposed and remembers how to fight off if you are exposed. So CRISPR is a mechanism in bacteria that allows them to remember when they were infected by a particular virus. So it acts like your immune system. And it uses that information to protect against future infections by actually taking a small bit of the viral DNA and inserting it into those regularly interspaced short palindromic repeats. Let's talk about how this works. So basically, this is a really, really brief description of this. You would start by identifying a gene of interest. So let's say that you want to edit the cystic fibrosis DNA gene. So cystic fibrosis is a genetic disorder, and it causes a lot of symptoms in individuals that are life-changing. These individuals have, well, we've talked about cystic fibrosis already, so we won't go into it too much. You guys know what that is. Basically, you would then use the DNA sequence to produce an RNA guide. So RNA is a molecule similar to DNA, and it is a coded molecule, so it is also complementary to DNA. So you use the DNA sequence to make this little RNA guide. So you put, using a plasmid, you put that RNA sequence that you want along with a sequence called the Cas9 sequence. So both of those are inserted into a plasmid. From there, that plasmid is inserted with something called a Cas9 enzyme. Now this Cas9 enzyme is a big molecule, and its job is to find the location in the cell where this RNA sequence is, bind to that sequence at the DNA, and make a cut. So essentially, it removes the chunk of DNA that is defective, or that you want to edit, or that you want to make defective. That's another possibility. You might want to study what happens when this is defective. And then, as part of this CRISPR system, the cool thing is where you have made that cut, you can now insert the sequence of DNA that you want. So you can put in a correct DNA sequence so you can repair damage, or you can knock out the DNA sequence. So you can put in a sequence that messes up, and you would do this to see what happens. This would be a research application. But if we're looking at cystic fibrosis, we would put in the correct DNA sequence that encodes the normal gene, not the disease gene, and then insert that into the genome of the organism in the target cell. So there are lots of really cool potential uses. One example is altering mosquito DNA to block malaria spreading, to block the pathways that actually help to spread malaria. Human trials are going on in which CRISPR was used in order to fight cancer. So it alters white blood cells so that they can attack and target cancer cells. Another example are these genetically engineered beagles to have more muscle mass than usual. And I'm not sure why you would do that. There probably was a reason. One of the first things that was done was in mice, and they were able to correct a gene for liver disease, in mice and actually get it into enough cells that the mice, basically the liver disease turned around. They were not able to eradicate it, but they basically were able to give mice a normal life. So there is definitely potential for CRISPR uses. But there are a lot of risks and potential problems for CRISPR. You can probably already imagine some of the risks that would go along with it. So there's the risk of legal issue. So there's already legal issues surrounding who invented this, who should get credit for it, who should profit from it, should anyone get profit from it, should it be copyrighted, things like that. And then there are also ethical issues. Should we be editing human genomes? Should we be editing human embryos? Should we edit basically defective DNA in an embryo? Now, on one side, you might say, yeah, absolutely. If you can give this person a better life, then go for it. On the other side, there's ethical issues. Is it actually ethical to do that? And then there's also the potential ethical issues with editing germline cells. So these are editing the DNA in sperm or eggs. Is it okay to do that? The other risk with CRISPR is that it's hard to predict the consequence of altering those genes. So you might look at one side of this example with not being able to block malaria spreading and saying, hey, if we can stop malaria, we can stop a lot of people dying. But the problem is that process actually kills mosquitoes. And if you kill mosquitoes, you're going to be taking out a food source for other living creatures in the area. And so those are potentially some unintended consequences that could have very different but also very dramatic effects on the environment where malaria is present. And then there's also sort of the unknown consequences, things that you don't even know are going to happen or that you can't even predict are going to happen. There are potentially unintended consequences to actually editing a gene. What happens if you're not successful? What happens if you don't get it to the right cell? What happens if you somehow editing that gene affects other genes? So although CRISPR is really cool and sort of cutting edge technology, there are a lot of risks and potential unknowns associated with it.